What if the most transformative medicine ever developed, one could cure a genetic disease with a single dose? Failed in clinical trials not because therapy didn't work, but because no one could measure it properly. That's not hypothetical, that's the reality facing dozens of cell and gene therapy programs right now. Hi, I'm Raghuveer Singh Parmar, Scientific Director at ResolveMass Laboratories, and today we are going to talk about why bioanalysis has become the hidden make or break factor in getting your CGT program from IND to approval. Why cell and gene therapy bioanalysis matters. Let's start with some context because I think it's easy to understand how different this space really is. For decades, bioanalysis was, was built around small molecules and monoclonal antibodies. You dose a patient, you sample plasma, you run your PK, clean, relatively predictable, well established. That's it. Cell and gene therapies break every one of those assumptions. You're not just tracking a small molecule that clears in hours, that you are tracking a living vector or a genetically modified cell that could persist in a patient's body for years, potentially decades. You need to know where it goes, how long it survives, whether it's expressing the right gene at the right level, and critically, how the patient's immune system is responding to it. And this market is not slowing down. Regulators like FDA, EMA are projecting 10 to 20 new cell and gene therapy approvals every year going forward. If you are working on an IND right now, this isn't a niche conversation, it's the central one. What is cell and gene therapy bioanalysis? So what does this bioanalysis actually involve? At its core, it's about answering four fundamental questions that regulators will ask you. Where does this therapy go? How long does it stay? Is it doing what it's supposed to do? And is the patient mounting any kind of immune response against it or not? To answer those questions, you are measuring everything from the viral vector itself to nucleic acid expression at the DNA, RNA, and protein level, all the way through to long-term biodistribution in tissues. It's not one assay, it's a program of interconnected analytical methods, each one needing to be fit for purpose, validated, and regulatory ready. Why this is a new frontier? Here is what makes this genuinely hard, and I want to be direct about this because I see sponsors underestimate it regularly. These therapies are biologically active. They behave differently from patient to patient. Interpatient variability isn't a nuisance, it's a defining characteristic of the data you'll generate. And to that fact that you are often required to follow patients for years post-treatment, and you quickly realize that the validation playbook from traditional bioanalysis simply doesn't translate directly. This is multidisciplinary science at its most demanding. You need molecular biologists, immunologists, analytical chemists, and people who understand clinical pharmacology, all working from a shared regulatory framework that frankly is still evolving. That's precisely why the choice of your bioanalytical partner matters enormously at this stage. Core components of CGT bioanalysis, where we look at the full analytical scope of a cell and gene therapy program, there are 5 major pillars: viral vector characterization, genome quantification, capsid analysis, empty to full particle ratios, transgene expression monitoring at the DNA, RNA, and protein level, cellular kinetics tracking, engraftment, expansion, and persistence of therapeutic cells, immunogenicity profiling, detecting anti-drug antibodies, neutralizing antibodies, and T cell responses, and biodistribution and potency, understanding whether therapy localizes and whether it's functionally active or not. Each of these requires a different platform, a different method, and a different regulatory justification. At ResolveMass, we have structured our cell and gene therapy service offering to address all five in an integrated way, because in our experience, the gaps between these pillars are exactly where programs run into trouble. Let's talk about regulatory landscape and challenges, because this is where many sponsors hit an unexpected wall. The existing ICH guidelines, they were written with conventional therapeutics in mind. They don't always map cleanly onto a therapy where your drug is a living cell or a replication competent vector. Regulators have been pragmatic about this. They understand the science is evolving, but that flexibility comes with an expectation of rigorous scientific justification. Potency testing, long-term follow-up protocols, comparability studies across manufacturing lots, vector setting assessments, all of these need to be addressed in your IND, and all of them needs bioanalytical backbone. The lesson we have learned working through complex regulatory submission is this: build your bioanalytical strategy before your IND, not after the first CRF is returned with questions. Key technologies in modern cell and gene therapy bioanalysis. Now let me get a bit technical, because the how matters just as much as the what. Digital droplet PCR, dt-PCR, has become indispensable for vector genome quantification. Its precision at low copy numbers is simply unmatched by conventional qPCR in this space. LC-MS gives us the specificity we need for therapeutic protein monitoring, including isoform differentiation that ELISA can't resolve. Flow cytometry is the backbone of CAR-T characterization, tracking cell phenotypes, persistence, and exhaustion markers simultaneously. And next generation sequencing is becoming standard for integration site analysis and genotoxicity risk assessment. These aren't technologies you want to develop assays on in real time during your pivotal studies. Platform familiarity and cross-validation experience matter. Future direction. Looking ahead, three things stand out. AI and machine learning are beginning to transform how we analyze complex flow cytometry data, moving from manual gating, which is a subjective and time-consuming, to automated reproducible pipelines. Decentralized testing models are gaining traction, particularly for multi-site manufacturing environments, and global regulatory harmonization, while still in progress, is moving toward more standardized expectations across FDA, EMA, and other agencies, which ultimately benefits sponsors with global development strategies. The window is to get ahead of these trends is now, not after your phase 2. Strategic importance of bioanalytical partnerships. This brings to me something I think is underappreciated in early stage CGT development. The type of partner you choose for bioanalysis can directly compress or extend your clinical timeline. A partner with deep CGT, cell and gene therapy expertise won't just run your assays, they will help you design a strategy that are regulatory defensible from day one, troubleshoot anomalies before they become deviations, and scale workflows as you move from phase 1 into pivotal studies. That's the difference between a vendor and a true analytical CRO partner. For small and mid-sized biotech and pharma companies, this is especially critical. You often don't have the internal infrastructures to build these capabilities in-house, and you shouldn't have to do that. The right CRO partner effectively becomes an extension of your analytical team, and that would save a lot of time and efforts. So let me leave you with the single most important takeaway from everything we have covered today. Cell and gene therapies represent some of the most remarkable science ever developed, but a therapy that can't be measured, can't be approved, and can't reach the patients. Advanced bioanalytical science isn't a supporting act in your CGT program, it's central to your regulatory strategy. It's central to your clinical decision making and ultimately your success. If your team is navigating any IND filing, early phase bioanalytical development, or just trying to make a sense of what your cell and gene therapy program actually needs analytically, let's talk. These conversations are exactly where the right guidance makes the biggest difference. Thank you so much for watching this session, and see you in the next one. Goodbye for now.